So the title, this is sort of a combined title. I think Jelena may have given me the title of Developmental Neurogenomics and the Neuroscience of Early Childhood Stress and Adversity. The subtitle, A Biology of Misfortune, is really the way I sort of think about how all of these different fields come together. That there are children in our midst, in our society, in our communities that experience unusual misfortune at the hands of the society's finances, at the hands of stressors and adversities that different children experience early in life. And the theme that I'm hoping to build through this talk is that there is now a science of that adversity and stress, that misfortune, that we are beginning to put together that will hopefully lead us ultimately toward an understanding of how we do something, how we intervene in the lives of those kids and those communities. So let me start here with this wonderful cartoon, a gene on the psychiatrist's couch, the psychiatrist saying, okay, let's go back to your childhood. So it raises issues of just how important are our genes. I think we are a field and a society that are absolutely transfixed by the genome at the moment. We have early evidence that genes in some sense remember the events of early childhood. And these kinds of issues raise these questions that have to do with how genes and environments work together to produce these varying trajectories of human development that I believe are very important, not only to children that we are rearing and watching being raised, but to our whole society as well. So let me offer as evidence of our society's preoccupation with genes and genomes this next little movie. Here is Tony Soprano, New Jersey mafioso boss, who in this little clip is speaking to his psychiatrist. But first, a warning that it contains coarse language not fit for civil North American society. So I hope you will understand as good Soprano fans the kind of language. But here you are what you are. How true is that, and where are we in this saga that we have followed over the last many decades of trying to figure out how social contexts and genes are related to each other, how they work together? Do we separate children and their genomes from the family contexts in which they operate and are being raised? Or do we begin, as behavioral genetics did a few decades past, to try to split the variance in outcomes that we care about into that is related to, that is attributable to genetics and that which is attributable to environments? Or do we begin looking, as we have in the last 10 years, at gene by environment interactions? Or ultimately, have we come now into a period of time where we're really talking about gene-environment interplay and the role of epigenetics in determining these kinds of developmental outcomes with which we are all concerned? So really within the professional lifetime of a single generation, as represented by your speaker today, there have been these extraordinary advances in understanding how social environments and constitutional vulnerabilities operate together to either produce or abate the risk of disease. So what I want to do today is to tell you two brief stories. The first is a story of how neurogenomic differences in susceptibility to environmental adversities amplify and diminish the risk for disorders of mental and physical health. And the second story is a story of how early exposures to social stratification and subordination may have lifelong influences on human morbidities. So let me begin with this first story about neurogenomic differences in susceptibility to adversity. So my work really began now almost 40 years ago as a pediatric resident at UCSF, which was my first vivid encounter with this phenomenon, which is the single most well-replicated finding in all of child health services research, which is that within any given population of children, there are going to be somewhere between 15 and 20% of the kids, represented by these little dark green figures, that represent well over half of the biomedical and psychiatric morbidities within that population, and are also responsible for well over half of the healthcare utilization that is carried out by that population. These children that are the majority, that have this smaller portion of the variance in morbidities and utilization, constitute about 80 to 85% of childhood populations, and it's about 15 to 20%, about 1 in 5, who are really responsible for well over half of the morbidity that we see within childhood populations. Now, the public health implication of that is obvious, right? It's that if we could figure out what is going on with this subset of children, this really kind of tiny slice of 1 in 5 children, we would be potentially accounting for well over half of the morbidity that we see within that population, and potentially able to do something about those children that are experiencing this kind of disproportionate morbidity. And when you begin to ask, well, why is that? What's the difference between these children and these children? What you begin to see over and over again, and returning to in the literature over and over again, is that socioeconomic status is the single most powerful variable that is attributable to these differences in these subsets of childhood populations. Socioeconomic status, if you think about it, epidemiologically, it is the single most powerful determinant that we know of mental health and physical health in childhood, and for that matter, adulthood populations. And yet, up until about 20 or 30 years ago, it was a variable that we never studied. We always controlled for it because we didn't believe any other association without first controlling for this variable. But we never studied socioeconomic status as a phenomenon and a force within itself. Now, in the last 2 to 3 decades, we have begun to do those studies, and some of what I'm going to show you today is the result of that work. So there is this socioeconomic partitioning, a social partitioning of child health and development. This is a figure taken from the work of Edith Chen, showing a variety of child health problems, including any limiting chronic condition, asthma, ear disease, injury, physical inactivity, as a function of socioeconomic status moving from lowest to highest. And characteristically, there is this fairly linear inverse gradient going from the highest morbidities in children in the lowest rungs of the social class hierarchy to relatively low levels in children in the higher reaches of the socioeconomic hierarchy. So poor children sustain systematically higher rates of acute, chronic, and disabling diseases. Now, what's maybe not most, but what's also very interesting about this is that this is not what my Canadian colleagues would call a hockey stick association. It's not that everything is level out here until you get to true poverty and then suddenly there's an increase in morbidity. It's that at every stage of socioeconomic status there is this graded increase, inexorable increase in morbidity as you step down the socioeconomic scale. And we don't really understand why that is. Do you have a question?
Because this is not a medical audience, can you define morbidity?
Yes, thank you. We use morbidity as a kind of global term that includes any kind of disturbance in health. So when I use the word morbidity, I'm including disorders of mental health. I'm including disorders of physical health, like what most of these are. So morbidity is a kind of global umbrella term that covers the span of health problems.
Thank you.
Sure. So this kind of mysterious graded association that is very powerful, always found in every society in which it's been addressed and looked at. And it's not only true that poor children sustain higher levels of morbidity within their childhoods, but it's also true that poor children have higher rates of cumulative chronic disease morbidity throughout their adulthood. So this is looking at coronary heart disease as a function of whether children were raised in low SES circumstances or high SES circumstances. This is a chart of cumulative morbidity, and you can see that as these individuals age, that there's a systematic and increasing distance between those who experienced relatively disadvantaged early lives and those who were raised in more advantaged communities. This is not just true of coronary heart disease, but is true of most of the chronic diseases of adulthood as well. Yeah, Michael.
Does that vary by the distance between quintiles? So in a place like the Netherlands, where the distribution is much flatter, do the curves look very different?
Yeah, they do. And I'm going to show some of that later on. This gradient looks very different in different societies. And we're going to talk about that in just a bit. So these conventional mediators that we know of risk for these kinds of chronic diseases, things like smoking, cholesterol, obesity, blood pressure, fail to account fully for this SES health association, leading to the question, what then are the mediators of these social disparities in child health and adult health? Well, we think that some of these mediators are issues that have to do with relatively mundane aspects of life as a child. This is just illustrative work. This is the work of Hart and Risley from 1995, who examined and really closely observed 42 Kansas families over 3 years of time. They virtually moved in with these families. And they found that when they looked at the number of utterances, the number of utterance communications between parents and children over time, that there were systematically fewer of these in the welfare families, slightly more in the working class families, and substantially more in the professional class families. So that by the end of the study, children in professional families had heard 11 million words in a year versus 6 million in working class families and 3 million in families on welfare. So that by kindergarten, if you total all this up, children from welfare families had heard on average 32 million fewer words than children from professional class families. So there are these systematic differences in the kind of stimulating, cognitive, enlivening atmosphere and environment of family life in different social class groups. It's also true that there is a socioeconomic partitioning of adversity and stress by social class. So that if we look at children growing up in impoverished families versus middle-income families, this is taken from the work of Gary Evans at Cornell, and we look at things like the density of housing, other housing problems, noise in the atmosphere, family turmoil, family violence. The exposures to all of these adversities is much higher in children growing up in impoverished circumstances than those growing up in middle income. Please.
Sorry, how is the violence in the indicator measuring? Why is it so high for families?
Why is the violence so high?
What is violence represented?
Well, in his work, he has looked at both intrafamilial violence, so marital conflict leading to violent conflict between parents or caregivers, as well as neighborhood violence, violence that happens outside of the family but within the community that surround the child.
Is the scale percentage?
No, the scale, no, it's not. This is a,
Scale.
yeah, it's a scale that Gary Evans used for monitoring. It's a standardized kind of scale for estimating differences in all of these different family characteristics. So the long and the short of it, and I've shown you only a sample of these differences, is that when you compare children growing up in disadvantaged to children growing up in middle class and upper middle class families, there are these just grinding, pervasive differences in the experiences of everyday life that occur every single day, every week, in all contexts, over and over again. Profound differences in the experiences in the first few years of life. But it is also true that there's a tremendous amount of variability in how developmental outcomes are related to these adversities and to socioeconomic status. So for example, here is a scatter plot of behavior problem severity scores by family stress in one of my early studies. And you can see that while there is a regression line that goes through these data points, it's highly significant. There's a tremendous amount of variability. There are kids who come from very high stress families who have almost no behavior problems at all, and kids who come from very low stress families who have lots of behavior problems that are sort of pre-syndromal psychopathology. Similarly, in Canadian data on literacy levels, I forget which is which here on these two y-axes, but both of them have to do with literacy and reading scores as a function of socioeconomic status. Again, you see a very strong monotonic linear association like we're used to seeing with socioeconomic status, but a tremendous amount of variability really at all levels of social class variation. So many years ago, we began trying to discern and think about what is this variability? Is this noise in the system? And if so, how can we clarify? How can we weed the noise out of the system by using better variables, better methods, better measures? Or is this really the music? And this is really what we should be looking at. And we decided 15, 20 years ago that at least part of the answer to that question is that this is at least partially music, and it is the part that we need to be attending to, the variability in how children respond to stress and adversity in their lives. So we began addressing this by looking at biologically the two most important salient biological systems in the human brain that respond to stress and adversity, the corticotrophin releasing hormone system and the locus coeruleus norepinephrine system. The CRH system begins here in the hypothalamus with the paraventricular and arcuate nucleus, which communicates down to the anterior pituitary, which releases ACTH. That goes through the blood to the adrenal cortex, which releases cortisol, which then comes back and shuts off the production of ACTH at the level of the pituitary. So this is the so-called cortisol system that we think of as one of the primary stress responsive systems within the human brain. The other is the LCNE system, which begins here with this little structure in the brainstem called the locus coeruleus, which has dopaminergic projections up to the hypothalamus, and which is responsible for igniting all of the fight or flight response that we're all familiar with. The dilation of pupils, the tremulousness, the sweaty palms, the increase in heart rate, and so on. So we began by looking at, if we were going to study the music, the differences between children and how they respond biologically to stressors, we would have to focus on these two systems. Why? Because these two systems have profound effects on target end organs that are important to health and well-being. CRH reactivity, which is shown here, has profound effects on glucocorticoid receptors, which then are responsible for decreasing the expression of pro-inflammatory immune cytokine signaling pathways. It also has effects on glucose metabolism, on cardiovascular homeostasis, and so on. The locus coeruleus norepinephrine system similarly has receptors on cell surfaces that's responsible for modulating the immune system, and also has effects on the heart rate on a variety of systems around the body. So we began bringing children into laboratory circumstances like this and studying individual differences in biological reactivity to psychological challenges. We had very highly scripted, highly precise, reliable paradigms that we put these children through, and we used measures of autonomic and adrenocortical reactivity to these standardized laboratory stressors using impedance cardiography to measure autonomic reactivity and salivary cortisol to measure adrenocortical reactivity. And what we found over the years was that there was a very broad and reliable individual variability in the magnitude and patterns of response within both of these systems. There were some children who in these circumstances were highly reactive in both systems, and there were many children, usually about 80 to 85% of the children, who had little blips in one or both of the system, one or more of the systems, but really minimal responsivity to the kinds of stressors that we were asking them to encounter. So now we took those variables of reactivity in the autonomic or the adrenocortical system, and we wanted to use those within epidemiologic study designs. And over a number of years, we have accrued studies that have looked at internalizing behavior problems, precursors, if you will, to anxiety and depressive disorders. We've looked at respiratory illness frequency and severity. We've looked at injuries that occur in children. And in one troop of rhesus macaques, we looked at violent injuries among members of the same troop. And finally, in a group of children, memory for stressful events. We divide the children up into those that in the laboratory look like high reactive kids versus those that are minimally reactive, and we look at them within naturally occurring social contexts that are categorized into those that are highly supportive versus those that are stressful. And these may be school environments, they may be neighborhood environments, or they may be family environments, depending upon the individual study. So I'm going to show you just a cartoon of the kinds of results that we have been consistently getting in these studies of reactivity and social context in interaction with each other. So we find, first of all, that the kids who have low reactivity profiles in the laboratory, when we look at them in different kinds of social contexts, there may be a little bit of increase under stressful circumstances, but basically there is a kind of indifference to the social context in which they are being reared. By the way, all of these children are, these are kids recruited in Berkeley and San Francisco. Most of them are children who are in very species-typical kinds of social contexts. None of these stressful environments were extremely severe. This is sort of a normal range within San Francisco and Berkeley communities that we're talking about here. When we look at the high reactive kids in the stressful circumstances, as we had hypothesized, these are the children who have more in the way of these undesirable maladaptive outcomes than any of the children within our study samples. But what we had not expected was that these same high reactive kids who are in very supportive low stress environments have the lowest rates of these maladaptive outcomes of any of the children in the sample. So we have a circumstance where we have a subgroup of kids who are phenotypically high in reactivity in standardized laboratory circumstances, who have either the worst or the best of the outcomes contingent upon the social context in which they find themselves. Now, there may be a number of different interpretations of that. The one that we first started talking about and have stuck with over the years is that this represents a kind of differential sensitivity to the character of the social world. That these are children who have either the worst or the best of the outcomes because they are the most sensitive to the character of the social environment in which they live. We used this wonderful idiomatic expression in Swedish, maskrosbarn, which means dandelion child, to refer to the low reactivity kids, the 80 or 85%, who, like the dandelion, seem to be able to grow and flourish in almost any circumstance in which you put them. These are kids who, at least within the bounds of fairly normal range of child rearing environments, seem to do pretty well irrespective of the kind of support or stress that they're experiencing. And we coined this Swedish neologism, orkidebarn, or orchid child, to refer to these children who, like the orchid, can have exquisitely wonderful, beautiful outcomes and lives, but who also, like the orchid, are very sensitive to the kinds of environments in which they are being reared. So dandelion children and orchid children, a simplification, especially when you consider the fact that the reactivity variable that we use to categorize these children is a continuous, normally distributed variable, but the orchid children are those at the very high end where things begin to look very different in terms of their outcomes by social context. Yes?
What are the experiments that test reactivity?
Sorry?
To measure reactivity, how is that tested?
The experiences that they went through in the stress paradigm.
Reactivity measurement.
That was the previous slide.
Previous slide.
Go back to
I think the challenges. I think it's the task challenges.
The task challenges. Okay, so they were ecologically representative kinds of challenges that kids in the 4 to 8-year-old age range would typically encounter. Being interviewed by somebody that you haven't met before, a stranger. Hearing a fire alarm go off in the midst of a laboratory session. Asking them to count backwards with digit span recitations. Asking them to look at emotion-evoking video clips that evoke various kinds of emotion. So these were all kinds of things that are not outside the normal kind of experience of children in that age group, but that generate a certain amount of perturbation in these stress response systems, which we can then measure and use in these epidemiologic study designs. Yes, Sean.
Is there any correlation between reactivity and socioeconomic background, or is it orthogonal to that?
There's some indication that kids growing up in low SES circumstances may have a higher representation of high reactivity profiles or phenotypes. There's a theory that we have that would account for that. Basically, that if you're growing up in circumstances that require vigilance and careful monitoring of the environment, that you may be more likely to develop a high reactivity phenotype because it's protective under those circumstances. But it's also true that it may, because of this phenomenon of these kids actually having lower rates of morbidity in supportive environments, we have also argued that there may be a higher proportion of kids with high reactivity phenotypes in environments that are exceptionally placid and supportive and routine and predictable. So we actually hypothesize that there would be a U-shaped distribution of the proportion of children who are high in reactivity by socioeconomic status. There's some preliminary evidence in favor of that U-shaped distribution, but it's not a lot. Pretty provisional.
Do you know how early this difference in reactivity develops?
My colleague, Abby Alcon, at the School of Nursing at UCSF, has been studying infants of farm worker families in the Salinas Valley. And she shows that even in the first year of life, you can see this differentiation into kids who are very high in reactivity in one or both of the systems, and most of the kids being relatively unreactive. So we think that it happens very early in life, and there's at least some evidence that you can detect differences in autonomic reactivity even during intrauterine life. So let me show you a little bit more of the kinds of findings that we've been getting within this sort of paradigm. So this is work that Nikki Bush has done, looking at the brain-derived neurotrophic factor polymorphism, the MET carriers and the VAL-VAL individuals as a function of family income looking at chronic daily cortisol levels. And she finds that those that are the MET carriers have either the highest daily cortisol levels or the lowest, depending upon whether they have one or the other of this genetic variant. So here again, the orchid children and the dandelion children. It even seems to extend beyond health outcomes and physiologic outcomes to developmental outcomes. So here is one study that we did with Bruce Ellis, looking at pubertal development, going from Tanner 1 stage to Tanner 5 stage. We all start here and we all end up here. And we do it over this period of time that is centered on about 12 and a half years of age, going forward 3 years and backward 3 years. And in this study, which was the Wisconsin study of families and work, we again divided children into those who were low in sympathetic nervous system reactivity. Those are going to be the ones shown in red, versus those that were high in sympathetic nervous system reactivity, which are going to be shown in blue, and then further divided them into those whose parents in observational work were shown to have high warmth in their communication and interactions with their children versus low warmth. So a four-way split: low and high sympathetic reactivity and low and high parental warmth. When we look at the trajectories of pubertal development in these children, the low reactivity individuals have pretty much the same kind of trajectory of development, whether they are low or high in parental warmth, a pretty linear trajectory going from Tanner 1 to Tanner 5. Again, everybody ends up in the same place, but this trajectory turns out to have powerful implications for risk for adolescent health. If you have an accelerated development, pubertal development, you're at risk for earlier sexual debut, the acquisition of sexually transmitted disease, adolescent pregnancy, and so on. So what do we find with the high reactivity individuals? Well, we find first that those with low parental warmth families have this accelerated development of pubertal staging, an accelerated move from Tanner 1 to Tanner 4 plus. Again, everybody ends up in the same place, but the children with high sympathetic reactivity and high parental warmth basically don't even go into puberty until about 12 and a half, and then finally catch up with their peers. So these different trajectories of pubertal development have great implications, and you can see that they are divided by both reactivity and parental warmth. The dandelion children, again, being those that have either the best or the worst outcomes, the orchid children, and the dandelion children being those that seem to be relatively indifferent to the effects of their kind of parenting. Please.
So I think you might have said this. So the high-low reactivity of the dandelion and the orchid, it's not above and below the median of the population. The orchids are the top 15% in terms of reactivity.
That's right.
And the remaining 85%.
That's right.
So
That's right.
So yeah. The males have some pretty strange behavior. If you look at the bottom 15% in terms of low reactivity, do you see sort of anomalous behavior there at the bottom?
The ones at the bottom are the flattest. The 85%, as I showed you, have this little bit of a tilt related to supportive or social or stressful social environments. But what you find is that when you get up toward the 80th percentile and above, there then begins to be this dramatic shift that gives us this orchid child sort of pattern.
So it's not a story of the outliers of 5%.
Not really. Not really. It looks like the outliers in this story are really at the top end of the scale. But mind you, even though we use this shorthand of orchid and dandelion, it really is a continuous Gaussian distribution. So we have evidence now for a neurobiological sensitivity to social contexts, both positive and negative contexts, that results in a subpopulation of children with greater variability in developmental and health outcomes. And there is a representation of this differential sensitivity at a variety of different levels, including autonomic adrenocortical reactivity, allelic variation, gene polymorphisms, and I didn't show you the data on cellular aging, but we also have data that suggests that these children are different in the level and the rapidity with which the telomeres at the end of their chromosomes shorten. Story 2, the health effects of social stratification and subordination. So one of the stories that I'd like to interest you in is this story of socioeconomic status, stress, and oral health. Why dental caries are the single most common chronic disease of childhood. There are strong socioeconomic and racial disparities. There are a lot of poor and minority children who have mouths that look like this by the time they start kindergarten. There are known etiologic factors that play into this difference: lead exposure, tobacco smoke, dietary differences, access to fluoridated water. And we also know that these bacteria in the mouth, these oral strep mutans and lactobacillus acidophilus bacteria, are also etiologic in the generation of caries in young children's teeth. And the leading account, the word on the street among most pediatric dentists, is that this difference between children of low and high socioeconomic status communities, the reason for the disparities in caries is a neglect of children's dental hygiene by low SES parents. We wanted to challenge that conventional wisdom because we also know, as I've shown you, that children from low SES communities also have far more in the way of adversity and stress, and we wanted to see whether that might somehow figure into this story as well. So this begins with us wanting to study children in the preschool and middle childhood years. In these studies, as I've told you, we typically collect saliva for salivary cortisol. And many of you probably know that cortisol secretion follows this kind of diurnal secretion patterns. It's highest in the morning right before awakening, and then it follows this sort of gradual decline and is lowest in the evening or right about when we go to bed. And for many years, we have been relegated to trying to understand the cortisol secretion patterns of children in our studies by sampling saliva at a variety of time points during this circadian rhythm. But the problem with that is obvious, that what we really want is the area under the curve. We really want to know how much cortisol exposure overall has this child had, not just what the cortisol was at 3 points in time during a single day or 2 or 3 days. So we began years ago to try to figure out how could we come up with a more chronic cumulative measure of cortisol exposure. And we started thinking about people with Cushing's disease, which is a pathological oversecretion of cortisol for endocrine reasons. The neuroendocrine system is, for reasons that aren't completely understood, is amped up, and the cortisol production is florid and overwhelming. And what these people get is demineralization of their spine. These are, you can see, collapsed vertebral bodies here. Here's one here, and here's one here. There's a demineralization of the bone that results in a variety of threats to health related to this kind of osteoporotic process. So we were thinking, well, what would be the analog of that in children? And obviously we're not going to do spinal x-rays of children to try to assess differences in cortisol exposure. But then we began to think, but wait a minute, we are actually studying individual human beings who are going to be losing a calcified tissue within the next year after our study began. Of course, all of these children that we were studying in kindergarten, in first grade, are going to start losing the teeth out in front. And so we began to propose maybe we could see demineralization in these deciduous teeth that are falling out of these kids' mouths and into our welcoming hands. So we began to think about the possibility that deciduous teeth could be a stress biomarker in young children, and in particular might give us more of a sense of the area under the curve for cortisol exposure. So we began doing computed tomography of the teeth that came out of these children's mouths. Here's a picture of one of these. It's just like a CT scan of a bone or a thorax, but it's a little tiny CT scanner, and we can estimate both the density and the thickness of these various layers of the microanatomy of the tooth. I should tell you that this arm of the study we called the Tooth Fairy Project, because we asked each of the children in this study to give us their teeth as they fell out, and we would reward them with $10. And that was way above the market value at the time of deciduous teeth under the pillow at night. And at one point, we sent all these teeth over to have them scanned at the dental school at UCSF. And one of them came back at one point in a little bag, and it said, "This is a dog's tooth." So somewhere in Berkeley, there's a dog that's missing a tooth because of an entrepreneurial kindergartner. So what did we find? Here's what we find. We found that when we looked at low versus high numbers of these oral karyogenic bacteria as a predictor of the number of decayed, missing, or filled dental surfaces, that there was this interaction with low versus high diurnal cortisol secretion. So karyogenic bacteria and salivary cortisol were independently and interactively associated with dental caries, the highest rates in children who had high karyogenic bacteria and high levels of salivary cortisol. Secondly, we found that when we looked at education level, low versus high as a measure of socioeconomic status, both cortisol reactivity and SES interactively were associated with enamel thickness. This is what's on the y-axis here. And the lowest thickness was found in children with high cortisol reactivity and low household SES. So putting these and a few other findings together, our understanding now of what may connect socioeconomic status and dental caries is two independent pathways, one of which goes through the proliferation and earlier introduction of karyogenic bacteria into the mouths of low SES children. And the second is a pathway that goes from low SES to higher levels of family financial stress, to greater HPA activation, greater cortisol secretion, changes in dental microanatomy as a consequence of this HPA activation, resulting in these two pathways leading to dental caries. And by far the strongest predictor of differences in dental caries was this interaction between karyogenic bacteria numbers and the level of cortisol secretion in the mouths of these children. So here were oral health disparities that seem to be related to two interacting pathways: earlier and more intensive exposure to an agent of disease among low SES children, the karyogenic bacteria, and more family adversity resulting in greater HPA activation, cortisol secretion, and microanatomic vulnerability. So what does all this mean, and what kind of a context can we put this into? And one of the issues that we have begun to study, and studied in fact in this same study that we studied the dental caries in, is the effects of hierarchy not just at the level of communities and societies, but at the level of these little micro-societies of children who come together within kindergarten groups. We know from Russ Fernald's data and studies that there are African cichlid fish that maintain severe hierarchical organization in which only dominant males have reproductive access to females. We know that rats in subordination paradigms show pro-inflammatory shifts in cytokine signaling pathways by the level of subordination or dominance that they are experiencing. We know in monkey species that these form stable, linearly transitive social hierarchies within troops, and we know that the subordinate positions within those troops are associated with upregulated adrenocortical function, impaired immune competence, and decreased resistance to disease. So the question then for us became, young children also form these kind of social orders within weeks of entering new social groups. So are the subordinate positions in these early peer hierarchies associated with greater stress, exaggerated reactivity, and excessive stress-related morbidity in the same way that we find those associations in the larger societal differences? So it was a question of, are these hierarchical relationships that we find within societies, if they are writ small in the hierarchical organization of young children in small social groups, do the same kind of health parameters layer as they do within the larger society? So we began to use within groups of kindergarten children in 29 classrooms, kindergarten classrooms in the Berkeley public schools. We began to study social dominance, and we defined social dominance as a pattern of repeated encounters in which the outcome consistently favors the same member of the dyad. We did dominance observations, which consisted of critical event and scan sampling of the classroom over 3 to 5 weeks with 2 RAs in each of the classrooms recording these events. And we recorded behaviors like physical attack, imitating behavior, directing behavior, threats, relational aggression. And we accrued a huge number of observations that resulted in about 38,000 dyadic encounters, which then at the end gave us the capacity via a computer software program to array these children from the top of the dominance hierarchy to the bottom of the dominance hierarchy within every one of the 29 school classrooms. You following me? So we're observing these kids, we're looking at these kinds of dyadic interactions between them. Let me just show you quickly what some of these look like. Obviously, none of these in and of itself tells us anything definitive about the relationship between 2 children or 3 children. But when these observations are accrued in the thousands over weeks of time, they begin to create a consensus about where children are within this hierarchy. You know, my medical students and other graduate students hate the fact that children of age 5 and 6 form dominance hierarchies. But in fact, this happens all the way down into the second year of life. It is very characteristic. Preschool and kindergarten teachers can tell you that within 2 weeks of children coming together in new social groups, they have a very clearly defined pecking order. So then the question becomes, what then? What are the consequences, or at least the correlates in terms of health outcomes of those different dominance positions? So at the level of societies, there is, as I've shown you before, this inverse association between hierarchical position and morbidity. The question is, in 338 kindergarten children and 29 classrooms, does the same kind of relation hold? And what we have found, and what we published about a year ago in the Proceedings of the National Academy of Sciences, is that subordinate children on the left here show higher rates of depression and inattention, poorer peer relationships, and poorer academic competence. It seems to be stronger for boys than it is for girls, but it actually is true for both boys and girls. So here is a pre-syndromal behavioral issue, depression, inattention, that is higher among those in lower subordinate positions than those who are in dominant positions. Now we're to Michael's question. if you look at these gradients of health by some measure of social class by nation, there's a tremendous amount of variability in the slope of those associations. So you can see here that there are places like New Zealand or the U.S. or Canada that have relatively steep gradients going from low to high SES. There's a fairly steep influence of SES on a variety of health outcomes. But there are also countries like Sweden, Switzerland, some of the Scandinavian countries that show flatter distributions. So there's variability in the slope of the SES development relation from nation to nation, and the flattest slopes are in the countries with these more egalitarian social policies. So then the question becomes, are there analogs to those social policies within these little micro-societies that we have been studying? And in fact, here is the source of the social policy within kindergarten classrooms. And here in a school of education, one of the findings that we have come away with from this is that kindergarten teachers create, this is no news to any of you in all likelihood, create cultures that are dramatically different in the way that they handle these hierarchical relations of dominance and subordination. We used variability in social position development gradients by classroom climate. We used the level of learner-centered practices, one of Debbie Stipek's measures, to characterize these different classrooms, because we had our RAs keep coming back saying, this classroom is very different from this classroom, we need to be measuring that. And we found that at the highest level of these learner-centered practices, which employed the most kind of egalitarian cultures, there was almost no relation between social position and depression. And here is this at a variety of different levels. So the ones with the most egalitarian cultures created by these teachers within the classrooms, there is almost no relationship between social position and teacher-reported depression, whereas in the low LCP classrooms, there was a profound and steep gradient. So the same kind of shifting of that gradient depending upon the kind of social policy enacted within that little society. Yeah?
I'm sort of surprised that such are used to thinking of like bullies who are dominant as being the ones, kids in the class who are the most trouble.
Mm-hmm.
But so yours is sort of the opposite of that, like, expectation. Like,
the kids who ended up in the dominant positions in our classrooms, the modal child, was, and by the way, I didn't mention this, but there was no difference by gender in the assignment of social position. The girls and boys were equally distributed in the full range of social position. But the kids who ended up at the top of the hierarchy tended to be children who were leaders. We would think of them as leaders. They were very effective at getting other children to do what they want, to engage them in play, and children deferred to them. And it was clear that most of the time the bullies, the kids that we think of as tyrants, were down at the bottom of the hierarchy, not at the top. So you're absolutely right. So we have here a conserved predisposition that seems to be across species to this kind of layered hierarchical social organization. And social subordination per se seems to be associated with diminished health and development, irrespective of the scale or complexity of the social context, going from micro-society to human society at large. And it also appears that at both levels of scale, that the consequences of inequality can be tempered by social climate and culture that is shaped by those who teach and lead. So I've told you two stories, one about neurogenomic differences in susceptibility, the other about the health effects of stratification and subordination. And what I was hoping to do was to show you how these two stories have converged in a third story of epigenetic neurobiological vestiges of early childhood adversities, which we're just beginning to be able to tell. But are we done?
We can continue. Since people have been asking questions along the way, we
Five more minutes?
Oh no, we ended at 7, so we have another.
Oh, okay. All right, great. Good. So let me tell you this third story. So one of the things that we have found in our studies of differences in social class is that there seem to be fundamental differences in brain development among children from low and high SES circumstances. So this is the work of Mark Kishiyama in the lab at Berkeley. He studied 8 to 12-year-old children from low, which are shown in the red lines here, and high SES backgrounds. He used a novelty oddball paradigm with this 64-lead EEG, 64-channel EEG. These are evoked potentials to stimuli that are presented in very controlled circumstances to kids of both social groups. He found that low SES children had reduced executive function performance, things like working memory, cognitive flexibility, semantic fluency, and so on. But he also found that there were these systematic differences in evoked responses by social class. Namely, the P1, N1, and N2 novelty components of these ERP amplitudes were concomitantly decreased in low SES children. And he concluded from this work that there seems to be altered prefrontal function in low SES children that may adversely affect their development and their ability to succeed in school and achieve academically.
Can you go back and tell me how to read the EEG line?
Yeah.
What am I supposed to get out of that?
Of course, electroencephalographers, up means negative and down means positive. So essentially what he did, these are average ERP responses to a visual stimulus over 1,000 milliseconds of time. So these are instantaneous kinds of responses of brain waves to the stimulus that brain is perceiving. And what he found was that there were these systematic differences when he looked at novelty detection in the low versus high SES children. These are actually topographic maps of the response in the high SES children for P1 and N1 and the low SES children. You can just see from gross inspection that there are differences in these. This is just a different kind of evoked potential, but again, the differences between the low and the high SES children.
And further away from the zero line is
Greater response.
better.
Yeah. So given that there seems to be this kind of developmental calibration of neurobiological circuitry, how might that happen? Well, we think that it may potentially happen in the same way that these two butterflies come out morphologically looking so different. They look like two completely different species in the overall color and patterning of their wings, but in fact they are the same species of buckeye butterfly, and this is in fact a polyphenism, a multiple phenotype that is driven by the temperature, the ambient temperature, and the length of daylight when they emerge from the pupal phase of their development. So this represents a conditional adaptation that involves differential epigenetic regulation of genes that determine coloration and patterning. They change their morphologic appearance to match most closely the kinds of, to optimally match the environment in which they are now emerging. So there must be some in this audience who don't know what epigenetics is. This is, I believe, an exciting new area of human biologic research. Just to give you a very quick primer, if you take a chromosome and unravel it, it turns out that the chromatin, as it is called, looks like beads on a string. This is the double-stranded DNA that is wrapped around a histone, a protein octamer, and this happens, it really is like a necklace of beads. And it turns out that the configuration of this chromatin can be altered by DNA methylation or other chemical alterations of the histone proteins that either opens up or closes down the chromatin, thereby allowing the enzyme that's going to decode that DNA to either function or not function. So experience can change the DNA methylation of a DNA strand, and that in turn can change the programming or the expression of that gene. So this polyphenism that I just showed you in the two butterflies seems to be driven by this kind of chemical alteration of the chromatin, this combination of DNA and in the nucleosomes. It is a mark that is placed there by the experience that fundamentally alters the expression of genes within the genome of the individual organism. So we have had one provisional chance to look at these kind of epigenetic changes in a longitudinal epidemiologic study of development. This was in Marilyn Essex' wonderful study of the Wisconsin Study of Families and Work, which began with over 500 pregnancies in the second trimester, and has followed these children now over 18 years to high school graduation. She's had like a 95% retention rate. There are wonderful developmental assessments all the way through the life course of these children and adolescents. And she has a variety of social contextual variables that were assessed along the way. In our case, we looked at infancy and preschool and looked at the stressors that had been reported by both mothers and fathers during these two developmental periods. Stress was operationalized as depression symptoms, expressed anger, parenting stress, role overload, financial stress, a kind of amalgamation of all of these various kinds of strains and stressors. We then followed up these children at 15 years of age, way down the pike here, using epigenetic profiling of buccal epithelial cells harvested from the lining of their mouths. We used an aluminum microarray platform interrogating 28,000 DNA sites in 14,000 gene promoters. And what we found is this. We found, and this was reported in 2011 in Child Development, that there seem to be these epigenetic vestiges of early developmental adversity as reported by mothers and fathers. So this is divided. This is showing differential CpG site DNA differential methylation. Above the line is hypermethylation. Below the line is hypomethylation. It shows it for the infancy period of the reported stress and the preschool period. The mother's stressors are shown in red and the father's stressors are shown in blue. And it shows it for all children, girls and boys, all children, girls and boys. So the summary here is that there is differential methylation of multiple CpG DNA sites by parental stress in both infancy and preschool. Mother stressors in infancy were more related to differences in methylation, and for both boys and girls, by far the biggest differences in methylation. Father stressors in preschool, that period, were associated with demethylation and primarily for girls rather than for boys. All of these associations were substantial in magnitude and had rows in the range of -60 to +60. So I'm going to finish here by arguing that this is one of the first pictures of human neurons that was presented by the great Spanish neuroanatomist Santiago Ramón y Cajal at the 1889 German Anatomical Society. He was using for the first time a stain from one of his colleagues, the Italian physiologist Golgi, and was able to show that the nervous system, the brain, was made up of individual cellular elements in the same way that livers are, skin is, and so on. But as important, that he was able to show that there were these points of connection between individual neurons, these synapses, which he presumed were the ways in which one neuron might communicate with another. The revelation of these connections and this cellularity of the brain led, of course, to what we now know as this at least pharmaceutical revolution in how we understand and can manipulate the functioning of neural circuits within the human central nervous system. And I want to argue that we may be living in the same kind of moment in the history of human biological science, where we are potentially discovering a nexus of gene-environment interplay in the form of these chemical tags that get placed on DNA and histone proteins and that alter the regulation of cells that fundamentally determine how we behave, personalities, and all of the kinds of individual differences in human functioning that we are concerned with. This is a drawing from an 8-year-old little girl who was a patient of mine years ago who had chronic abdominal pain. And she gave me this as she left the clinic one day. Here she is, right here. And down at the bottom she says, I got left out when the big kids teased me, and my heart is like breaking. Here's a message that was left on the internet by Tim Kretschmer, a secondary school student in Germany, posted on the internet the night prior to shooting and killing 15 people in his school. He said, I'm sick of this messy life, always the same. Everyone makes fun of me. No one recognizes my potential. I am serious. And then finally, in the maybe most current and widespread version of this, the Occupy Wall Street movement, where really an entire generation became motivated to express their concern for the non-egalitarian, layered character of the human society in which they are living. What all of these different, very different little vignettes have in common is experiences of social stratification, subordination, inequality, stress, and adversity. And I want to leave you with the notion that social relationships are actually very important. And they are particularly important during periods of economic descent, greatest amount of income inequality in this country since the Great Depression. We are living in very uneven society in sociometric terms. So I believe that, and I will end here with this favorite line from Ernest Hemingway, the world breaks everyone, and afterwards many are strong in the broken places. I think it really is our needed commitment and one of the most important things that we can do as educators, as physicians, as psychologists, as developmentalists, to try to find the ways of making those who are broken within our societies, particularly those who are young and broken, strong in those broken places. So I will stop there. Yelena?
We have about 10 minutes for questions. If you could, Tom, repeat them just for the recording, that would be great.
Sure. Yeah. Maybe they've already been asked.
Yeah.
Yes.
I have a question. The measurement of cortisol in America, how reliable is that? I know it comes up in many studies. I'm not a scientist myself, so I think that, but I've been told that it's not that reliable. So I need to clarify that.
It can be variable, and it's very responsive to serum cortisol. So in terms of its validity, there's good evidence that it is a valid reflection of the cortisol that is in the blood at the time. But like the cortisol that's in the blood, it also can fluctuate at least from minute to minute, from time epoch to time epoch. So there is a fair amount of variability. All the more reason for needing some kind of a cumulative measure of cortisol over time so that we get some kind of a picture of the general activation of the cortisol system over a longer period of time, not just a momentary estimate at individual points in time. I should also tell you that, I mean, what we did was a fairly elaborate and technically arcane way of looking at the effect of cortisol on the density and the thickness of these micro layers in deciduous teeth. But there are other cumulative measures of cortisol that are now being studied and used in studies, like hair cortisol. Doesn't hurt to take a clip of hair. It turns out that our hair typically grows about 1 centimeter per month. So particularly in people with long hair, you can actually estimate what the cortisol level was at different points along that time frame. If you have an 8 centimeter sample of hair, you can estimate cortisol at 8 different points in time. And it is cumulative. It is a much more stable and, if you will, reliable measure of cortisol exposure. Saliva is less so. Yes?
Following the discussion of the hyper and hypermethylation, and what that meant in preschool. Looking it up on Wikipedia, but I was trying to figure out. So you're saying that these gene markers are actually turned on and off still after they've been born and into preschool.
Yes.
Is that something that can happen throughout a lifetime, or does that end at a certain time? And kind of what are all of the genes kind of open for different expressions of that age? How does that work?
Yeah, one of the real revelations in the last 10 to 15 years has been the fact that it was previously thought, and I'm going to rely on Russ Bernald here to, as the baloney detector. If I'm saying something that isn't true, will you tell me? It used to be thought that the epigenome, these marks, these chemical marks that were responsible for up or down regulating the expression of genes were pretty established very early in development and probably didn't change. One of the things that has happened in the last X number of years is the revelation that these things do change, that there are epigenetic marks that come and go with circumstance and with experience. We don't know on what kind of a time scale, as far as I know, whether it's, minute to minute, second to second, day to day, what the sort of time scale is. But what is clear is that there is plasticity within the epigenome and that it is responsible to the kind of experience that we have. Originally, we thought that the epigenome was only for taking the same genome that's in every one of the cells in our bodies and using that genome to create liver cells versus skin cells versus neurons. And that obviously is epigenetically mediated because it's the same genome. We have 200 different kinds of cells in the body. How do you get 200 different kinds of cells out of the same genome? You turn things on and off. And so it's fairly important for a hepatocyte, a liver cell, to remain a liver cell and to divide and produce more liver cells. So that has been known for a long time. What is more novel is the idea that there is this kind of plasticity in epigenetic profiles that comes from the experiences that we have within physical and social environments. Everything there true?
Yeah, I think that one of the difference now is that we know that the epigenome can change in hours, so that their shortest data are 2 hours, and that recently it's been shown that there can be these DNA methylation marks at non-CPG sites. So it's now possible to find them lots of places in the genome and alterations that were really previously unsuspected.
Yeah.
Tom, do you want to clarify that the epigenetic stuff was done at 16 years or 18 years, and so the just connection to the stressors are different?
Yeah, I mean, what we thought was remarkable about this finding was that there was a decade and a half between the report of the stressors and what we were measuring as epigenetic differences in mid-adolescence in these same kids who had been little infants and toddlers at the times that their parents had reported the stress.
Sorry, just about, you say to change in hours, and then we're saying to measure a decade. later. What does that mean? What are the two differences there?
Well, you have one genome, and it has to do lots of different things. It does it by turning on and off a piece of itself. Those inactive genes, for example, that replace neurotransmitters, can be altered on a very short time scale. The longer permanent time scale is the one that Tom described, which sets up the developmental programs for different cell types.
So there are probably different time scales for different kinds of
But we
changes.
have studies showing that if you're taking a really stressful test during the duration of the test, that there's some methylation happening too, right? In a response to that experience. So it could be that stress can also work on a different time, right?
So I hope I've adequately conveyed the excitement of this because, we've debated this nature-nurture thing for, decades of time. It's the big biological dilemma that Waddington first tried to solve by saying that there was this thing called epigenesis. What these epigenetic marks and these changes in the chemistry and the conformation of chromatin are potentially representing is where the genome and the environment actually come together. It's the actual physical place where things can change in a way that change the development of the brain, change the response to stressors and adversity, all of that. Yeah.
I was interested on the study on subordination. I was wondering if there were any links between the propensity for the child to be subordinate to if he's an orchid type or dandelion type, assuming that the orchid type being more sensitive would be more likely to. Or I don't know if that relation exists or if it exists in both directions or only for one.
Stay tuned. There are a lot of analyses of that data set that we haven't yet undertaken. Thankfully, Jelen is helping me with these analyses, and there will be some kind of an answer to that question. I don't know yet. Anecdotally, my guess is that there are going to be more of what I've called the orchid children toward the bottom of the hierarchy relative to their presence at the top of the hierarchy, but truly stay tuned. Do you want to say anything about that?
Yeah, I do. It ties in a little bit about who the dominant kids are. We're finding that reactivity differentiates our leaders from our bullies. So dominant, especially in boys, dominant boys who are highly reactive are our bullies who are fighting to get that to that dominant spot. And our dominant boys who are low reactive are liked by peers and liked by teachers, and they're very pro-social and are the leader type. So it could be that there's different pathways that you can achieve dominance, and that may be reflected in your physiology.
So try to imagine a masculine orchid. That's the sort of mix that we're involved with here.
I wonder if you ever probed those children to know if they have an internal representation of the status that you perceive. In other words, can you imagine finding out from them how their rankings match yours?
Oh, yes. Let me see if I can remember this. When we looked at the observation-based assignment of social position, it was moderately but significantly associated with teacher estimates of where children were in the ranking, but not the child's estimation. The child's estimation was basically orthogonal. So it's as if the children don't really know where they are in the hierarchy, but an adult observer from the outside seems to have at least a moderate level of ability to predict where a child is in an observation-based assignment of social position. Yes? Yeah.
So there's a controversy in the equality literature about whether outcomes depend on the actual variance in wealth versus just sort of the absolute amount of poverty. Do you know what I mean? So for example, some people would argue that if everyone's getting richer, even if the difference between the rich and the poor is increasing at the same time, that that's a good thing, whereas other people would say that is the fact that the rich are getting richer compared to the poor is a bad thing. It seems that your research sort of bears on this question. In, like, for example, in the hierarchies, like just comparing yourself to somebody who's higher than you can have a negative outcome, even if absolute, like in terms of the mean, everyone is higher. You know what I mean? Would you say that's something that you're...
Well, the thing that we have found interesting in this layering of health outcomes by social position within kindergartners is we started off, by the way, to try to find multi-pan socioeconomic kindergarten classrooms in Berkeley. And we couldn't find any. So we're not able to test the hypothesis that kids from low SES families tend to go to the bottom of the hierarchy and vice versa. What did I start out to say? So what we find remarkable about this is that these were all within classroom kids of basically the same socioeconomic status. It's a homogeneous classroom economically. And yet, even within that homogeneous environment, there are these subtle differences in teacher and parent and child-reported health that vary by social position. So even in the absence of money, even in the absence of differences in the trappings of wealth within the larger society, the fact that dominance and subordination seems to have, I'm not going to say it doesn't necessarily cause, but it is associated with differences in health status. So I think that's at least part of what you're getting at.
Yeah, it's interesting to consider like just relative wealth. I mean,
Yes.
scaling it up.
Yeah.
You can't directly infer it, but just comparing yourself to somebody else.
Yeah.
Even if your mean across the population has gone up, just
Yeah.
by virtue of the variance also increasing, that could have negative health outcomes.
Yeah.
Yes.
We have never really had a dialogue with a health economist about these data, and I think that would be a very interesting thing to do. Yeah.
Do children ever move from high reactivity to low reactivity? And if so, how?
If you look at test-retest reliability of reactivity status by age, Abby Alcon's work that I mentioned in the Salinas farm children, in infants and young children, shows a significant test-retest reliability. So there's some tracking of reactivity. If you move up to middle childhood, in the study that was most of what I was showing you, there's an increase, a bump up in that level of reliability. It's even more replicable from one time to the next. And then if you go up into adolescence, Karen Matthews' work at University of Pittsburgh, she shows really quite robust test-retest reliability in reactivity status. So it seems like there's sort of a cannibalization of reactivity as you move up in development. It becomes a deeper trough and harder to get out of. Now, that's not to say that there isn't variability within those various trajectories, and I'm sure there are children who over time could look high reactive at time 1 and low reactive at time 2. I'm just talking about in sort of general terms.
Have they been ever associated with interventions?
In reactivity interventions. You know, there have been a few studies of people trying to use like hypnosis to change the reactivity status of individual, and I think in children as well. But, you know, from where I'm coming from, this reactivity status is not necessarily what needs to be changed. It's more of a marker for the vulnerability of the child, the susceptibility differentially to the social environment. So I'm not sure I would necessarily target the reactivity itself as something that would in the long run make some kind of a difference in the trajectory and outcome for an individual child.
I'll have to cut off. We're out of time at this point, but thank you once again.
Thank you.